few questions in biology and developmental biology that is very important to us. And one of the main questions that we are asking is how to go from one fertilized egg cell to a whole organism. So fertilized egg divides and generates multiple cell types and tissues, and finally an organism. And there is iCloud coming on. Shall we try to just, how do I?
It's turned off.
It is turned off?
Yeah.
Okay. Yeah. And in our lab, we are interested in how this is controlled at the level of transcription. And protein coding genes are transcribed by polymerase II, and transcription is controlled via chromatin modifications, like Peter showed you. If you would just think about DNA as this black line here, which is packed with nucleosomes, we have chromatin modifying enzymes that is doing a lot of job putting repressive marks, activating marks throughout the genome, and this will lead to either silencing of genes, or it can lead to recruitment of polymerase to a gene that's going to be transcribed. For example, here we have a DNA binding transcription factor that could recruit an enzyme. This enzyme is a cofactor that removes a repressive mark, which is the lysine 9 trimethylation here. Next, we can have a compass complex, which have a trithorax protein that will put K4MA3. This mark is recognized by hat activity complexes such as this Saga complex here, that will, via GSM5, generate an acetylation. This is an acetylation on lysine 14. This can promote binding of general transcription factors such as TF2D, and we will get recruitment of polymerase. Once polymerase is recruited to the gene, it will start to transcribe the gene, and as Peter explained many times at many genes, polymerase is paused right downstream here at the transcription start site. And the kinase complex PTFB is then recruited down to polymerase and can phosphorylate the polymerase and phosphorylate negative elongation factors that is released from the polymerase. And polymerase can then move into a productive elongation. When polymerase is elongating, factors such as the SET2 enzymes tethered to this tail CTD structure of polymerase and distribute K36 trimethylation throughout the gene. In this case, KDM4A can here remove the K36 trimethylation, but normally K36 is believed to be important for the inhibition of insertion of acetylated histones into the gene body. So K36 trimethylation is then inhibiting spurious transcription of this gene. I divided this into three different parts. The first part will be about PTFB kinase. Second part will be about KDM4A enzyme. And the third part is about histone 3 lysine 14 acetylation. So first part, PTFB, the superelongation complex, and mediate or regulate non-paused, rapidly transcribed genes during early Drosophila embryo development. So here we're interested in studying PTFB and its role in early transcriptional regulation. And PTFB consists of two different proteins. It's an enzyme that is phosphorylating polymerase, and it's cycling. The polymerase is stalled here in a pause position downstream the transcription start site. And the hypothesis that we have is that PTFB will come in and release polymerase from paused genes. And to do experiments in early embryos, we need to use UAS-GAL4 system here to generate microRNA-mediated knockdown of maternal factors such as PTFB already in the ovaries. So the female have ovaries where she produces her eggs. The eggs are filled with maternal factors such as PTFB, and mRNA can here be produced by us in the ovaries to deplete PTFB. So we will generate embryos where PTFB protein is depleted. So when we do that, we can see the depletion of PTFB subunits, CDK9 or cyclin T, result in developmental defects. So this is a control embryo with its head and its tail. And these are either CDK9 embryos or CDK9 has now been depleted from the embryo, or cyclin T embryos. And we can see here, compared with control, that this depletion results in severe morphological defects. Next, we could rescue this phenotype by introducing back a CDK9 transgene. This transgene is containing a microRNA-resistant sequence sitting in this exon. By doing that, we can rescue PTFB phenotype. So our hypothesis was that PTFB would regulate genes that have post-polymerase. So next, we were interested in looking into this by screening a number of different early patterning genes, where some of them have post-polymerase. And we first needed to know that we had a knockdown that was sufficient in our embryos. So we monitored either a flag-tagged version of our CDK9 to see that we can knock down this construct, looking at control compared to the knockdown. And also monitor the amount of cyclin T knockdown. You can see here that we have a reduction in 2 of the cyclin T isoforms. So now we are starting to screen different genes in the early embryos. And these are 4 different patterning genes. You can see here these control embryos that these 4 genes are expressed very nicely in different patterns. The genes are being regulated upstream by a number of different factors. Although in the PTFB depleted embryos, these patterning genes are still expressed in rather normal levels. However, in the posterior part of the embryo, we have a phenotype, which is interesting. It's marked out here by the blue arrow. And this was a novel phenotype to us. We wanted to investigate what this phenotype is about. And to do that, we stained embryos for membrane and for nuclei. And here in CDK9 and cyclin T depleted embryos, we can see that in the posterior part of the embryo have lost all its cells. So PTFB depletion resulted in a loss of posterior cells. We also saw a cellularization defect, which is this image here. So looking down on the surface of the embryo, apically, the normal wild-type honeycomb pattern here with membranes surrounding each nuclei is distorted. We have now several nuclei within one cell, and so this is now a phenotype called cellularization defect. Next, we wanted to investigate why or when this phenotype, the posterior phenotype, is happening. So we did this by introducing a fluorescent nuclear marker, which is a histone that is tagged by an RFP. In this way, we can monitor the embryos by taking pictures of the embryos every second minute, and then make a video of this. These are wild-type embryos. Here you can see that we have a very rapid nuclear division that we have nuclei that is covering the whole embryo, and this now turns into a gastrulating embryo. This is the head furrow here. We can run that again because it looks so nice. So here we're starting to fill the whole embryo with dividing nuclei. From the beginning, remember that we just have one fertilized nuclei. So very rapidly, this embryo is filled with 6,000 nuclei. So let's see what's going on now in the P2P embryo. So this is CDK9 depleted embryos. We have embryo filling up with nuclei, just like in the control. It's just prior to gastrulation what is happening. So here we see a loss of cells in the posterior part of the embryo. And this embryo is not going to survive. So here clearly see that the embryo first have nuclei throughout the embryo, and then later these posterior cells are lost. Next, we wanted to investigate the cellularization phenotype. We know from the literature that there are several zygotic genes that are involved in cellularization. So by checking some of those genes, we could see that the gene serendipity alpha is downregulated in the P2P embryos. However, serendipity alpha is a gene that have no post polymerase. It's a gene where polymerase is transcribing the gene without pausing at the promoter. So this was in contrast to our hypothesis. Next, we found another gene called terminus. Terminus and its duplication, CG7271, also have a reduction in expression in P2P embryos. Terminus is an unknown gene, not characterized before. So this led us to try to characterize it a little bit by either doing a knockdown of the gene or overexpressing the gene product. So here we see that in the knockdown and overexpression, we have cellular defects in the embryo. So this concludes that terminus is important for early development. Other genes that are known to be involved in cellularization are bottleneck, SLAM, and nullo. So we now focus on these three genes. What we could see is that neither of bottleneck, SLAM, and nullo have any different expression in P2P embryos controlled to wild type. So here we have a P2P regulate a small set of genes that seems to be non-post. So next we wanted to look at one of the substrates of P2P, which is phosphorylated polymerase. So here we have used antibodies that recognize either the polymerase itself or phosphorylated polymerase. Now we can look at the amount of phosphorylation per polymerase. So here we have P2P target genes, such as serendipity alpha and terminus, and they show a reduced level of phosphorylated polymerase. However, non-target genes also show a reduction in this phosphorylated polymerase. So, since P2P is known to interact with polymerase by releasing it into elongation, we speculated that removing P2P might stall some of the polymerase in a pause position, and thus change the ratio of polymerase in the 3' to 5' end of the gene. So we would, in that case, get higher levels of polymerase in the promoter than in the end of the gene. But this is not the case, because when we compare the wild type ratio to P2P ratio, 5 to 3', we see that the polymerase ratio is unchanged. However, what we can see here is that using the polymerase antibodies, we see that at P2P target genes, we have a decreased occupancy of polymerase itself. This is not the case at P2P non-target genes. conclusions of the first part, in vivo P2B depletion result in a novel posterior loss of cells phenotype and cellularization defect. A subset of non-post genes are extra sensitive to reduced P2B levels. P2B depletion does not turn non-post genes into post genes. P2B depletion leads to a reduced POL2 SERIN2 phosphorylation levels at most genes, suggesting that absolute POL2 phosphorylation levels are not critical. The decreased POL2 occupancy at target genes suggests a role for P2B in POL2 recruitment, or might indicate a secondary effect due to failed POL2 promoter release. And with this, I will go into the next part, which is called gene regulation by the lysin demethylase KDM4A in Drosophila. So as I previously showed you, with the gene and the nucleosomes and everything here, the enzyme KDM4A can remove this repressive mark K9 trimethylation. It can also remove this mark that is distributed during POL2 elongation. In order to study KDM4A, we generated flies that are mutants for this gene. So here we can see control flies, and here is the relative expression level of this gene KDM4A that is producing this KDM4A protein. This is compared to the mutants that we produced that have very, very little expression from this gene. We can rescue the expression levels by introducing a transgenic KDM4A gene or a transgenic KDM4A gene that is having a mutation which generates a catalytically dead enzyme. So one of the substrates for this enzyme is histone 3-K36 trimethylation. Next, we tested if we have any changes in the global levels in larvae. So here we have normal conditions, control larvae, and comparing that to the mutant larva, we see that we have an elevated level of this methylation. When we are putting back the transgenic KDM4A gene, we are down to normal levels of this methylation again. However, when we are putting back an enzyme that is catalytically inactive, we are having an elevated global level of K36 trimethylation. Next, we overexpressed the same enzyme, and now we saw that the enzyme is regulating these levels in the opposite direction. So more of the KDM4 enzyme will generate larvae that have a less K36 trimethylation level. So KDM4A controls global H3 K36 trimethylation levels. Next, we generated RNA, and we're interested in looking at if KDM4A is regulating genes. And this is validated by qPCR, so we can see here that the red is control gene expression, whereas the blue bars are the same genes have less expression in the KDM4A mutant. You could also find genes that are upregulated in the KDM4A mutant. Looking at the same genes when we're overexpressing KDM4A, we see the genes that previously went down in the mutant now have a higher expression when we are overexpressing KDM4A. The other genes that went up in the mutant are now going down in the overexpression larva. Next, we wanted to investigate the catalytical function of the enzyme and try to see if the function of the catalytic domain is important to regulate these genes. So here we can see that we found genes that are at the level of 1 in the control larva, and in the mutant they are very lowly expressed. When we are putting back the transgene of KDM4A, we can get back the levels of these target genes. However, when we're putting back a transgene that is carrying the point mutation, we also get back the expression levels. Other genes are dependent on the catalytic activity. Here we have two genes where we can see that putting back the catalytic dead enzyme in dark green does not rescue the levels of this gene back to normal. So not all KDM4A target genes are regulated by KDM4A's catalytic activity. So next, since we knew that KDM4A is regulating K36 trimethylation levels, we wanted to see that if these genes are regulated by changed levels of K36 trimethylations. So doing chip experiments with an antibody that is recognized this specific histone mark K36 trimethylation, we could see that in wild type larvae, the levels of K36 are very low. However, in KDM4A mutant larvae, the levels are also really, really low compared to control locus. Another substrate for KDM4A is K9 trimethylation. Here we have the same thing, that we have very little K9 at the target genes compared to another control locus. So KDM4A target genes have little histone methylation. Finally, we found that when we're increasing the levels of KDM4A by overexpressing it, we see a male lethality. So here we are comparing the number of males and females that are going all the way from embryo up to adulthood. You can see that we have only 6% males surviving. So to conclude this, KDM4A regulates global K36 trimethylation levels. KDM4A regulates gene expression in vivo, possibly through a histone demethylation independent mechanism. Overexpressed KDM4A is male lethal, suggesting that it is involved in a dosage compensation mechanism or specifically regulates sex-specific genes. So my next part number 3, histone 3 lysine 14 is essential in Drosophila. So here we have the same chromatin model again, where we have MLL trithorax protein distributing K4-ME3 mark. This mark is recognized by factors with HAT activity that can acetylate K14. It's been shown that in yeast that JHD2 enzyme can demethylate K4 trimethylation. But if we have K4 acetylation, this will inhibit the enzyme from working. So in this way, we will build up the levels of K4 and K14. So we are now interested in studying the K14 acetylations and its role in development and gene regulation. We do that by making histone replacement flies. So we will create flies here that have no wild-type histones and only have containing histones that have a K14 mutation. This is a project that contains flies that have multiple transgenes, and it might be a bit complicated for people not doing genetics, like mom and dad. I will try to explain it. So here we have a histone deficiency on second chromosome that will take out all wild-type histones. We use this in combination with multiple histone genes that we're cloning together in a big construct. And these histone genes, the histone 3, have a substitution where we substituted here, so we have K14 arginine. And this is then put back into the fly on the third chromosome. In this way, we can generate flies that lack the wild-type histone locus where the bulk histones are sitting, and only have a K14R histone fly. So selecting larvae that are mutant for the wild-type histones, we can see that here we have homozygous for the deficiency and only 12 copies of the K14R. Here we have their siblings, these larva siblings, they are heterozygous for the deficiency and they have 12 copies of the K14R replacement. These are larvae that are heterozygous for the deficiency, and instead of having the substitution, they have 12 wild-type copies. Here we see that we have much reduced levels of K14 acetylation. So, next, we stained embryos with this antibody that recognize this mark. So here we have embryos again that are heterozygous for the histone deficiency, and they have 12 copies of the replacement histones. And here we can see in the central nervous system that we have this K14 acetylation. In these embryos' siblings that are homozygous for the deficiency, we no longer have any acetylation in the CNS. To conclude this, K14 acetylation levels are diminished in H3K14R larvae and late embryos. Since we saw that these embryos and larvae that only have the replacement, they do not survive until adulthood. So we decided to pick and test at what stage these embryos or larvae die. So here we have homozygous deficiency that have 12 copies of wild-type replacement histones. Here we have the numbers of surviving individuals after 124 hours. These are flies that are homozygous for the histone deficiency and have 12 copies of the replaced histones that have K14R. So both of these different strains have no wild-type histones, they only have the replacement. And here, the larvae that have K14R do not survive after 124 hours. And here is the same numbers plotted in a different way. This is the control. These are the K14R larvae that all are dead at this stage. Next, we decided to do a clonal analysis, and this is to see how cells that lack wild-type histones behave in other tissues. So here, we decided to put an FRT chromosome together with the deficiency, and we also introduced a heat shock-induced flipase, and then we have the replacement transgenes on another chromosome. And what we do is that we can generate mitotic clones by a heat shock. So here we can see that in the clones where we don't have any wild-type histones, they are marked out by absence of GFP. The other control cells have GFP. Now we're dissecting out tissues, for example, wing discs. This is a tissue that later in the adult flies are the wings. So here we can see a twin spot where we induced a mitotic recombination event, which have led to proliferation of many cells. So here we have control cells, and next to them are cells that lack wild-type histones and only have the replaced histones. So looking at the darker staining, clearly see that the cells that lack the wild-type histones and only have the K14R histones are proliferating. Staining wing discs with a K14 acetylation antibody, we see here that we have no longer any acetylation in cells where we have induced this recombination. If we are letting these embryos that we are inducing the clones, if we let them go to adult flies, we have some of them surviving. And here we could see that K14R mitotic clones result in adult phenotypes similar to a GSM5 hypomorph, GSM5 being an enzyme that can distribute the K14 acetylation. Finally, we decided to look into this mechanism which was shown in yeast, that K14 acetylation inhibits JHD2 from demethylating K4 methylation. So this enzyme is called LID in Drosophila. So what we did was to stain embryos for K4 trimethylation. So these are the control embryos that have one copy of the deficiency, and they also have wild-type histones, so they are heterozygous for the deficiency, and they have 12 copies of the replacement. Here we have staining in the CNS, whereas in their siblings doesn't have any wild-type histones, no longer have any K4 trimethylation. So H3K14 acetylation regulates K4 trimethylation levels in vivo, possibly via inhibition of LID and its demethylation activity. To conclude this, K14 acetylation is essential in Drosophila, but K14 acetylation is dispensable for cell survival and proliferation, suggesting that K14 acetylation controls specific gene regulatory pathways instead of controlling all genes. K14 acetylation regulates K4 MS3 levels in flies, suggesting a conserved mechanism in yeast and multicellular organisms. And with this, I would like to finish this by saying that our data point to the importance of using model organisms, such as Drosophila, where it becomes clear that in vitro data cannot always be extrapolated to in vivo systems. The molecular mechanisms in vivo are many times far more complex than in a test tube setup. For example, we revealed a novel phenotype resulting from P2B depletion, and we found a role for P2B in the activation of non-pulse genes in early development. We also showed that KDM4A might regulate genes by a different mechanism than via its histone demethylase function. These findings were unexpected and would have not have been possible to achieve using in vitro systems. In addition, we showed that it is possible to generate multicellular organism devoid of K14 acetylation. The advantages of using genetic tools is that it's possible to reveal biological complexity and not discovered by pure biochemical assays. And with this, I would just like to thank you for listening.
Now we do like a talk show. So Olle, as I promised in my introduction, so in your title, you mentioned the super elongation complex, but in the talk that you just gave, you didn't mention that at all. Can you tell me a little bit about the super elongation complex? What is it? And why do you think it's part of the story?
Yeah, so super elongation complex, P2B kinase core complex is a major or a component of the super elongation complex. However, P2B is also found in together with other complexes. The super elongation complex uses P2B for gene activity, and flies have one copy of each gene that uses proteins in the superelongation complex compared to humans that have multiple copies of each of the subunits. It's known that mediator can recruit the superelongation complex together with P2FB and thus activate genes. So in that case, it might bridge mediator and P2FB and put P2FB where it should be at the polymerase. Also, what we found was that different subunits of the superelongation complex is giving